This, again, like the topic we did last week, photosynthesis, it's very similar with the fact that GCSE, we've been told basically just really simple word and formula equation for what respiration is. You know that there's aerobic, there's anaerobic, and that's sort of where it ends. What we're going to actually look at is the actual molecular processes of this now, actually in detail, how it works in detail, and look at that a lot more. So it is a very complex process, and again, it's in lots of cycles and lots of chain reactions. So again, in a very similar way to what I said about photosynthesis, the way I would suggest revising and learning respiration is to learn it in stages, to write down the cycles, write them in chains, do them as diagrams and learn them, write them, have a go writing them again, try and write them from memory, then copy and have a look, where did I make those errors? Get your one that you've done from memory, and then get the one that you've got your perfect set of notes, compare them, add the information through, and keep doing that again and again. And personally, I think that's the best way. I think the hardest thing about respiration and photosynthesis is that they are both very similar, but they are completely different processes. So what can often happen is sometimes people in the head can go, oh, is this the Kelvin cycle? Is this the Krebs cycle? And get a bit confused. So not only is it important to know both of them in a lot of detail, it's really important to go, okay, this stuff and information is part of photosynthesis, this is part of respiration, and know the difference that way as well. So what is respiration? I'm going to go super back to basics to start off with. So respiration is that process by which organisms extract the energy stored in complex molecules. And we use it to generate adenosine triphosphate, ATP. ATP is that energy currency molecule. We're extracting energy from complex molecules. So ATP, we don't use it as a storage molecule, but organisms will store energy in the form of complex molecules, things like glucose, starch, other complex carbohydrates, and then we extract the energy from those complex molecules and convert it into ATP to then be used as that current useful energy currency right then and there. So this is how ATP is basically how all metabolic pathways are fueled. And this is the way that the organisms obtain that fuel, is they store it and then convert it. This is ATP. You'll have seen it a lot last week when we were doing photosynthesis. Here it is again. It's a huge part of respiration. So ATP, it provides the immediate source of energy for biological processes. So it's used for things such as active transport, movement, metabolism, anything like that, really. So as we said again, we've got the three different parts of ATP present. We've got our three phosphates, we've got our adenine, and then we've got our ribose sugar. So we have got two types of respiration. We've got aerobic respiration and we've got anaerobic respiration. So in aerobic respiration, so that is respiration that takes place in the presence of oxygen, a respiratory substrate. So what we mean by this thing, a respiratory substrate, is one of those complex molecules. So respiratory substrate, for example, glucose, it's split in the presence of oxygen to release carbon dioxide and water, okay? Aerobic respiration produces a large number of ATP molecules that are released from that energy from the glucose. So aerobic respiration produces a large amount of ATP. Opposite, anaerobic respiration does not produce as much ATP as aerobic. So here is our equation for aerobic respiration. So we've got glucose, C6H12O6, oxygen. So glucose plus oxygen provides us with carbon dioxide, which will be our waste product, water, and then obviously ATP. And here, this is one molecule of glucose. So someone's got one molecule of glucose and 6 molecules of oxygen. We make 6 molecules of carbon dioxide, 6 molecules of water, and 36 molecules of ATP. That's really important to remember. So this is where we're saying we make a large number of ATP from one molecule of glucose when it's going through aerobic respiration. Conversely, anaerobic respiration, this is when glucose is split and it's converted to either lactate or ethanol. In the absence of oxygen, produces a very, very low ATP yield. So what happens here is glucose becomes either ethanol or lactate. In this example, it's ethanol, carbon dioxide, and this time, for every 1 molecule of glucose, we're only producing 2 molecules of ATP. And same process when it's lactate, 1 molecule of glucose only produces 2 molecules of ATP. So it is not as energy efficient. Really useful when it's needed, but again, it's another reason why we cannot rely on anaerobic respiration all the time. So there are 2 types of respiration. The whole point of respiration is to basically make ATP. So where does respiration happen? Where does it occur? So respiration occurs in every single living cell possible. In eukaryotic cells, the early stages of respiration occur in the cytoplasm, so the cytoplasm of the cell, and then the later stages of respiration are restricted only to the mitochondria. So when we talk about mitochondria, we say it's site of respiration for synthesis of ATP. So early stages of respiration can occur in the cytoplasm of the cell, and then later stages are restricted just to inside the mitochondria. So a bit of a quick recap of mitochondria. These are organelles that are found inside eukaryotic cells. Mitochondria have this really intricate, highly folded inner membrane here. Why is it highly folded? It increases the surface area. It means there's more sites for respiration to occur. So these highly folded inner membranes hold key respiratory proteins, so things like the enzymes that we need to make ATP, over a large surface area. Mitochondria provide an isolated environment to maintain optimum conditions for respiration. So just as photosynthesis needs optimum conditions, so does respiration. Mitochondria basically protects the environment and maintains that it's in the correct optimum conditions so that respiration can be constantly ongoing at the same rate. Mitochondria have their own DNA, remember, and they have ribosomes, so they can manufacture their own respiratory enzymes. That's really, really useful. So basically, we're not relying on other cells or other organelles to do that, but mitochondria are able to do that all themselves. So, quick recap of parts of mitochondria. So we've got this outer layer, which is our outer membrane. We've got this inner folding going on, and that is the inner membrane. So it's got an outer membrane and then a folded inner membrane. Then got this blue substance inside, and as we can see on this image here, got it there. Oh, no, wrong image. And that is our cytoplasm. It is the matrix that's going on, the matrix inside the membrane. What we're pointing at here. This is the cristae, folded, the actual infolds here. So that is the cristae I think we are pointing at, or is it pointing at the ER? Can't actually tell. Give me a second and I'll work that one out. That is definitely pointing at the ribosomes, so that makes that easier. And this one is obviously going to be pointing at the DNA loop. Right, is it pointing at the highly folded cristae or the ER? I'm going to go with highly folded cristae. We'll find out. Yeah, there we go, the highly folded cristae. So, mitochondria, again, how much do we know about them? Mitochondria are the cellular organelles in which majority of respiration occurs, okay? The inner membrane is folded many times to form structures known as cristae. So that inner membrane that's folded, think back to GCSE, we call that inner folding of the membrane. They're known as cristae. And these cristae provide a large surface area. Again, that thing in biology that we always go back to. If we've got folding, such as like in villi, in cristae, it's for providing a large surface area. It increases the rate of reactions. So these provide large surface area for the later stages of respiration to occur. Mitochondria contain their own circular loop of DNA, which codes for the many enzymes used in respiration. And these are constructed by the mitochondria's own ribosomes, because obviously that's where proteins are made, which are found in the matrix. The outer membrane ensures that the conditions inside mitochondria remain ideal for respiration. So, respiration. We're going to now break it down into lots of stages, make it a little bit more complicated. Both aerobic and anaerobic respiration start off exactly the same. So regardless if we're talking about aerobic respiration or anaerobic respiration, they both start with glycolysis. And this is the first stage of respiration, regardless of if we're talking about aerobic or anaerobic. So that is glycolysis. Now, glycolysis occurs in the cytoplasm of the cell. Okay, so it occurs in the cytoplasm of the cell. It's not necessarily actually going to occur inside. It's not going to occur inside the mitochondria. This is happening in the cytoplasm of the cell. And the point of glycolysis is it converts glucose into a molecule called pyruvate. And it produces a very, very small amount of ATP. And no oxygen is needed for glucose. What actually happens is glycolysis actually breaks down glucose, and it doesn't just make one molecule of pyruvate, it makes 2 molecules of pyruvate. Now, this process is exactly the same for anaerobic or aerobic. But what ends up happening is we end up making something different at the end. So I'll explain that in a second. So glycolysis will make pyruvate, can be used for anaerobic or aerobic. In anaerobic respiration, this is obviously going to occur with oxygen. And what happens is those products of glycolysis, that pyruvate and the tiny amount of ATP, are then broken down, and it allows glycolysis to continue. Okay, so they're broken down, glycolysis can continue, and in anaerobic respiration, so we've got no oxygen, products of glycolysis are broken down and it allows glycolysis to continue. So it can either produce lactate or ethanol and carbon dioxide, and we'll look into that equation in a little bit more detail in a moment, because that's a little bit different for each one. Why is this not moving? In aerobic respiration, oxygen is used, so that pyruvate is broken down to regenerate ATP. And there's actually 3 stages to aerobic respiration after glycolysis. So really there's 4 stages. So for aerobic respiration, we have glycolysis. We then have something called the link reaction, the Krebs cycle, and then this one's called 2 names depending on where you read it. This one says ETC, which is electron transport chain. Other places, the AQA textbook refers to it as oxidative phosphorylation. So it just depends what textbooks you're using. That's 4 stages. And we're going to basically just do a really quick overview of the 4 stages, where they happen, what happens, and then we're going to look at them in detail. So the link reaction happens straight after glycolysis. The link reaction, this is all the parts that are going to happen actually in the mitochondria. So this occurs in the mitochondrial matrix, and it evolves some carbon dioxide. So glycolysis is where glucose goes to pyruvate. We add on the oxygen, link reaction happens inside the mitochondrial matrix. After the link reaction, we have the Krebs cycle. In the Krebs cycle, it produces a number of reduced coenzymes. We'll look into them in a bit more detail in a moment. And this also happens in the mitochondrial matrix and involves some carbon dioxide along with a small amount of ATP. And then finally, we have the electron transport chain, or sometimes referred to as oxidative phosphorylation. And this uses reduced coenzymes to regenerate lots of ATP. And this occurs in the inner mitochondrial membrane and converts oxygen and water. So that was a super fast overview of the beginning of this. What we're going to do is look at those molecules for respiration in a bit more detail, because there's some molecules that we haven't really talked about before, and there's some that we have. So ATP, and I have a super fast overview of ATP because we've looked at this loads recently. ATP, it contains that sugar ribose, a base, which is adenine, and 3 phosphate groups. So we've talked about hydrolyzing ATP a lot. So when we add water to ATP, we can produce ADP and an inorganic phosphate. And this is basically what happens when we're releasing energy from ATP. So when we do this, when we hydrolyze ATP, 30.5 kilojoules of energy is actually released. So we get quite a lot of energy being released per ATP molecule. That's quite an important bit of information to know. So why do we use ATP? So biological systems transfer the energy in glucose to ATP because, unlike glucose, ATP is able to release energy instantly in that single reaction. So in that hydrolysis reaction, that 30.5 kilojoules of energy is just released instantaneously. That doesn't happen in glucose. And the hydrolysis of ATP releases a small amount of energy, and that's ideal for fueling those reactions in the body instantly. So phosphorylation of ADP. So this is obviously how we make ATP. So that addition of that inorganic phosphate to a molecule like ADP, we call it phosphorylation. And this happens a lot during respiration. And we have 2 types of phosphorylation occurring during respiration. If you think back to photosynthesis, we talked about photophosphorylation a lot. Obviously, that's not going to occur here because we're not relying on light. So we have 2 different types. So we have 2. We have substrate level phosphorylation. And this happens in glycolysis in the Krebs cycle. And this is when a single reaction involves a direct transfer of a phosphate group from a donor molecule to ADP. And I'll highlight when this happens. Now, basically, we have lots of intermediate molecules involved, and you don't have to know them. All you have to know is that it's substrate level phosphorylation. So it's a single reaction. So basically, phosphate is being donated from one molecule to the ADP. That's all you have to know. And I'll point out when that happens. You don't need to know the names of the molecules that it comes from, the donor molecules. You just need to know that there was one there. And then there's also oxidative phosphorylation, which occurs in the electron transport chain, or sometimes we just call it oxidative phosphorylation. And this is the more complicated phosphorylation. So this is where we have a series of oxidation reactions that produce sufficient energy to form ATP from ADP and phosphate. So that's an overview of why the different types of phosphorylation of ADP. Something that we would have talked about a little bit in the past when we've talked about enzymes is a coenzyme. So if we think back, enzymes are a biological catalyst. They speed up the rate of reactions. We have an active site that's got a specific shape to a substrate. Now, a coenzyme, as you can see in this image here with that little orange part attached to the enzyme, is a molecule that's able to bind with a specific enzyme or substrate. Okay, so coenzymes, if we think back to when we've talked about enzymes before, and I'm happy to recap this in the future if we want a bit of a recap. Coenzymes can either actually attach themselves to the enzyme or to the substrate, and they help catalyze a reaction. So actually they can speed up a reaction even more. They actually can help catalyze the reaction even more. So sometimes we need that coenzyme as well. Breaking the bonds between a coenzyme and the product after a reaction is crucial. Otherwise, what happens is that coenzyme concentration will drop and it limits respiration. We use 3 different major coenzymes in respiration. So one that we've actually come across before, and we came across it in photosynthesis, that's the word, is NAD, which is nicotine amide adenine dinucleotide. So NAD is used. CoA, coenzyme A, and FAD, which is flavine adenine dinucleotide. So all 3 of them are coenzymes that we use during respiration. So NAD, let's look at this one again. This one was in photosynthesis. NAD is that one that we reduce a lot because it can accept a hydrogen molecule and it forms reduced NAD, which we write as NADH. So NAD can accept a hydrogen molecule, forms NADH plus that extra hydrogen molecule there. It's also a reversible reaction and we can then oxidize NADH to make NAD. We use this to regenerate ADP in the electron transport chain or in oxidative phosphorylation. So we regenerate ADP in this manner. We also have coenzyme A, and coenzyme A helps us to transition between glycolysis and the Krebs cycle. So glycolysis is that first stage of respiration, and then we have the link reaction, and then we have the Krebs cycle. And coenzyme A helps that transition because what it does is it converts pyruvate to a molecule called acetyl coenzyme A. And that is what we need to start the Krebs cycle. So coenzyme A helps us transition between glycolysis and the Krebs cycle. And finally, FAD, like NAD, can accept hydrogen to form reduced FAD, FADH2. So what do these molecules do? Coenzyme A, it aids in the transition from glycolysis to the Krebs cycle. NAD plus is a hydrogen acceptor. Short-term energy store of a product respiration is ATP, and inorganic phosphate is moved between molecules to vary their energy levels. So first stages of respiration then, glycolysis. I'm going to get something different up actually now. I'm going to show you this differently. So, here we go. So the first stage of photosynthesis, don't know why I said that. The first stage of respiration is glycolysis. And it's really important to remember glycolysis is the first stage, regardless of whether we're talking about aerobic respiration or anaerobic respiration. So glycolysis is the first stage, oops, made itself smaller, regardless of whether we're talking about the first stage of respiration, aerobic respiration, or we're talking about anaerobic respiration. So glycolysis is this first stage. And remember, it's, let's pop it here. So we've got glycolysis. Glycolysis is that first stage. And the point of it is it converts glucose into 2 molecules of pyruvate. And that is the point of it. Okay, so that's what I'm going to go through now, is the stages of glycolysis. And that is identical for if we're talking about aerobic respiration or anaerobic respiration. So here we go. We start off with our molecule of glucose. Now I'm going to write on how many carbons each molecule has. And you'll understand why in a bit. So I'm just going to pop this on. Glucose is a 6-carbon molecule. And the first part of glycolysis is actually phosphorylation. So I'm just going to pop a little number 1 over here. The first part of glycolysis is phosphorylation. This whole thing is glycolysis, and this first part is phosphorylation. So we start off with glucose. We've got a glucose molecule here. And our glucose molecule is going to be phosphorylated. And it's going to be phosphorylated by an inorganic phosphate that's come from ATP. So what happens is we have ATP. ATP goes to ADP. Oops. And the inorganic phosphate here phosphorylates our glucose. Okay, so glucose is phosphorylated by the inorganic phosphate that's come from this ATP here. So, like we said before, ATP, it can get broken down and we can have ADP and an inorganic phosphate. That inorganic phosphate phosphorylates our glucose and it forms this molecule. And this molecule is called glucose phosphate. Glucose phosphate is a 6-carbon molecule. What then happens is exactly the same thing. The glucose phosphate is phosphorylated, and this forms a molecule called hexose bisphosphate. Hexose bisphosphate. And that's done it exactly the same way from the ATP. So our ATP is broken down. And we've got ADP. And our inorganic phosphate is there. That's been added on. Hexose bisphosphate, again, 6-carbon molecule. The last part of phosphorylation is our hexose bisphosphate is split into 2 triose phosphate molecules. So now we've got 2 lots of triose phosphate. Now, I'm going to write 3C here. That's because each triose phosphate is a 3-carbon molecule. Okay, so the hexose bisphosphate splits into 2 triose phosphate molecules, and each triose phosphate molecule is a 3-carbon molecule. And this whole section here is phosphorylation, because we've got phosphorylation here, and it splits. The second part of glycolysis is oxidation. That's what I'm going to go through now. So our 2-triose phosphate is oxidized, and it becomes 2 molecules of pyruvate. How this happens is this way. I'm just going to do a different color pen just to show this easier. So oxidation is loss. We know that. So that means that we've lost some hydrogen. So what happens here is that 2-triose phosphate loses 2 hydrogen ions, 2H plus. And what then goes on is we've got that NAD, that nicotine adenine enzyme, coenzyme. So NAD comes along, it collects that H plus, oh, it's NAD, collects that H plus and is reduced to form 2 lots of reduced NAD. Okay, so that's what happens, oops. When that triphosphate is oxidized, that's what happens to the 2 hydrogens. Okay, so they go over there, reduces the NAD, we get 2 lots of reduced NAD. And that was obviously 2 lots of NAD being reduced. Let's just make that clear. Then we also produce 4-ATP. And what happens as well here is we've got 4-ADP, 4 lots of, that's AP. We've got 4 lots of ADP and 4 inorganic phosphates. they are going to make 4 molecules of ATP. So 4 molecules of ATP are produced. Now, if we look up back here, we've used 1 molecule of ATP here, and we've used 1 molecule of ATP here. So our net gain of ATP during glycolysis, net gain of ATP equals 2. So we've only gained 2 ATPs in total during glycolysis because we've used 2 in the earlier stages. So if we carried on with aerobic respiration, our NADH that we've made is going to be used later in oxidative phosphorylation, oxidative phosphorylation. That's going to be used later in that. And our pyruvate would go via active transport to the matrix of the mitochondria and would be used in the link reaction. So that is what happens to those molecules if we were carrying on with aerobic respiration. And obviously that ATP can then just get used up. So the product of glycolysis is, I'm going to say 2 ATP, because obviously we've used 2 earlier, so our net gain is ATP is 2. So we've got 2 molecules of ATP, 2 molecules of pyruvate, and 2 molecules of reduced NAD. So what is happening then if we are actually going to do anaerobic respiration? So let me clear this a second. So we've gone through all of this. I don't want to clear the whole thing, so let me just put my eraser on. Let's just get rid of this for a minute. Let's get rid of the stuff that tells us about aerobic respiration. Doing anaerobic. Go back to anaerobic for a moment. Okay, so what actually happens? Well, we know the product of anaerobic respiration is either ethanol or lactate. So let's have a look at what happens. So what happens is, if we go through anaerobic respiration, what happens to that pyruvate? Well, that pyruvate is broken down. We either get ethanol or lactate. So this is what can happen. We could have pyruvate. Pyruvate. It can be broken down to ethanol. When it's broken down to ethanol, it's I can never say this word, carboxylized, so it loses its CO2. Becomes ethanol. Then it becomes ethanol by being oxidized by NADH, reduced NADH, and becoming NAD. So it's oxidized to make ethanol. Or if we're making lactate, pyruvate, can become lactate. Oops, I might need a bigger arrow. Lactate by being oxidized. And that is the process of what then happens, and then anaerobic respiration stops there. So if we go back, if we're going to go through aerobic respiration, we are going to go on to stage 2. So stage 1 is glycolysis. Happens for aerobic and anaerobic respiration. The second stage then is something called the link reaction. Now remember, ATP is now going to go off and get used elsewhere. NADH is going to be saved for later for oxidative phosphorylation. So we'll come back to reduced NADPH, phosphorylation, but we're going to use that pyruvate straight away in the link reaction. So, link reaction. What is a link reaction? Where does it occur? What is happening? Oops. So, number 2. Link reaction. Okay, so the link reaction occurs twice per glucose molecule. So this is good to remember. It occurs twice per glucose molecule. It's really important to remember that. So this process will happen twice for every 1 glucose molecule we're breaking down. So glycolysis happens once, and then link reaction will go through twice, okay? And this happens in the mitochondrial matrix. Mitochondrial matrix. Now remember, pyruvate that we're going to start with has got here via active transport. So pyruvate enters via active transport. Okay, so let's talk about the link reaction then. So we're going to start off with pyruvate. Pyruvate is a 3-carbon molecule. And this is why the link reaction happens twice. Because remember, we've made 2 molecules of pyruvate in glycolysis. So therefore, the link reaction has to happen twice per glucose molecule because it happens once for each pyruvate molecule. So pyruvate is a 3-carbon molecule, and pyruvate is decarboxylated. So it has its carbon dioxide removed. And again, I'm going to put my carbons on again. Oops. It is a 1-carbon molecule. So our pyruvate is, I want to make that longer as well. Our pyruvate is decarboxylated, and it is also oxidized. And it is oxidized by our NAD, and we form NADH, so reduced NAD. So remember, oil rig oxidation is lost, it loses a hydrogen that is picked up by the NAD. And when pyruvate is oxidized, it makes the molecule acetate. Now, acetate is a 2-carbon molecule, and that adds up and makes perfect sense because we've lost a carbon here when the pyruvate was decarboxylated. Now, acetate. Acetate is going to eventually form this molecule called acetyl coenzyme A. Coenzyme A. To do this, acetate and coenzyme A join together. So let's add in coenzyme A over here. Coenzyme A comes in. And we get acetyl coenzyme A being formed. And again, acetyl coenzyme A is a 2-carbon molecule. Now, really, really, really important to remember, this happens twice per glucose molecule, once per pyruvate molecule. It happens in a mitochondrial matrix. The pyruvate has entered via active transport and no ATP is produced. Okay, so at this point, what we should make then is we've got 2 lots of acetyl coenzyme A. That's actually right, probably. Coenzyme A, and this will go into the Krebs cycle. So it's going to be using the Krebs cycle. we have got 2 lots of carbon dioxide, which is going to be waste. And we have got 2 lots of reduced NAD, which is going to be used in the oxidative phosphorylation, along with that original reduced NAD back in glycolysis. So we've got that one that's going to get used there, those 2 there, and we've got these 2 that are going to get used as well. So that is the second stage of respiration for aerobic respiration. So actually, if I just added another one here, the stages of respiration, aerobic respiration, are glycolysis, link reaction, Krebs cycle, and then, depending what you call it, yeah, I've got electron transport chain or oxidative phosphorylation. Exactly the same process, just 2 different names. Glycolysis happens in the cytoplasm of the cell. Link reaction happens in the matrix of the mitochondria. So they are the 2 that we have done so far. Now we are going to look at the Krebs cycle. Krebs cycle, you're going to see it, and it's going to probably be very, not familiar, but very reminiscent of, what is the word I'm looking for? The Calvin cycle. But yeah, Krebs cycle. It basically is a series of oxidation and reduction reactions. And again, this one takes place in the matrix of the mitochondria. And this happens once for every pyruvate molecule. So this happens twice per glucose molecule. So, Krebs cycle. This is stage 3. I'm going to write it here. Oops. Just driving mad that you doing that. Krebs cycle. So happens twice per glucose molecule because it happens for each pyruvate molecule. So I'm going to talk it through with you. First thing that happens is, let's go back and look at what we made here. So link reaction, actually let's go back to beginning. Glycolysis. Start off with glucose. Glucose is phosphorylated to different molecules until we eventually get to triose phosphate, where it's oxidized and it becomes pyruvate. Pyruvate actively transported to the matrix and goes through the link reaction to become acetyl coenzyme A. So pyruvate is decarboxylated. It is then reduced. It then has coenzyme A added to it, and it forms acetyl coenzyme A. And this acetyl coenzyme A goes into the Krebs cycle, which is what we're going to look at now. So we've got acetyl coenzyme A, and we have got a 4-carbon compound. This 4-carbon compound is called oxoalactate, spelled O-X-A-L-O-A-C, oxoacetate. Acetate. There we go. So what happens in the Krebs cycle to begin with is acetyl coenzyme A and our 4-carbon compound join together. So acetyl coenzyme A comes in, joins with that. Now, when acetyl coenzyme A joins with our 4-carbon compound, let's put our carbons on, like we like to do, that is a 2-carbon molecule. It then releases the coenzyme A. So coenzyme A can then be released. And then that goes back to the link reaction to get used again. So coenzyme A is recycled constantly. Okay. So at this point, this goes back to link reaction. When these two molecules join together, they form a 6-carbon compound, which makes sense. 4 plus 2 equals 6. And this 6-carbon compound is called citrate. And just so that we're marking our carbons as we go in, 6C here. Okay, so our 6-carbon compound is going to become a 5-carbon compound. So let's talk about how that's happened. So our 6-carbon compound is going to become a 5-carbon compound. So it's converted to a 5-carbon compound. How this happens is decarboxylation occurs first. Okay, so we have decarboxylation of the 6-carbon compound. So that is CO2. And that is removed, which obviously is a 1-carbon compound. So that is why we've gone from a 6 to a 5-carbon compound. But we also have dehydrogenation occurring, and that is where a hydrogen is removed. So that occurs like this. So that NAD that we like comes along, removes the hydrogen, and becomes reduced NADH. Okay, this is really important because we're going to use that reduced NADH again later. So 6-carbon compound is decarboxylated, so we can get the 5-carbon compound. It's also goes through dehydrogenation, so that hydrogen is removed. The hydrogen is used to produce reduced NAD from that. Okay. What is going to happen now is our 5-carbon compound, is eventually going to be converted into our 4-carbon compound. But as you can probably guess, a lot of things have to happen for that to happen. I'm going to just draw this a little bit smaller there so we can then have space to write. So 5-carbon compound is going to be converted to a 4-carbon compound, which is that oxaloacetate acetate molecule. It happens through the decarboxylation and dehydrogenation of different molecules. So it happens reducing FAD and NADH. So I'm going to go through each one and what happens. So decarboxylation happens first. So we've got the removal of carbon in the form of a carbon dioxide molecule, so CO2. So let's track out carbons again. That's a 1-carbon compound. So again, 5, remove 1, 4-carbon compound. Okay, so then we've got lots of dehydrogenations occurring. So first one is the same as the ones that we've done before. We've got NAD. It's going to accept the hydrogen that's removed, and that becomes reduced NADH. Then we're going to produce some ATP. ATP is produced by the direct transfer of an inorganic phosphate from an intermediate compound. So this is that substrate phosphorylation we talked about before, where it just takes one straight away. So you don't need to know the name of the intermediate compound. But this is what happens basically. So what happens is an intermediate compound donates that inorganic phosphate, which joins with the ADP. So then we get ATP. Then I'm going to run out of space. This will work. Then what happens is we've got dehydrogenation again 2 more times. And the first one is going to be of FAD this time. So we're going to lose a hydrogen and it's going to be accepted by FAD. So then we get reduced FAD, FADH2. And then dehydrogenation occurs again, and this is going to be NAD accepting it, becoming reduced NAD. So this, remember, I'm going to just highlight that, is substrate level phosphorylation. Then, as you can probably work out, this 5-carbon compounds become 4-carbon compound, and then the cycle can happen again because the 4-carbon compound is going to join the acetyl coenzyme A. So remember, this happens twice per glucose molecule. So this is molecule. This is happening twice per glucose molecule. Our list of products are going to be, that's one I haven't used. We're going to have 2 molecules of ATP. We're going to have 2 lots of CO2. 3 lots, no, not 3, no, my maths has gone wrong there. If we're doing it twice, we're going to have 4 lots of CO2. We are going to have 1, 2, 3, 6 lots of reduced NAD, and then 2 lots of reduced FAD. So that is what our products are. Okay. That is a Krebs cycle. What happens to those products? Well, I'm going to go through what happens to those products now. Oh, and obviously we've got coenzyme A actually as well. I'd forgotten that. So I'm just going to go through quickly with you what happens to all these products. So this happens twice. So again, quick talk through. Acetyl coenzyme A that was produced back in the link reaction comes along and binds with the 4-carbon compound oxaloacetate. Coenzyme A is then released, and that can go back to the link reaction. And the joining of those 2 compounds forms a 6-carbon compound, which is called citrate. This 6-carbon compound is going to be converted to a 5-carbon compound. It does this through 2 stages. The first stage is the decarboxylation occurs, so we lose a CO2, a form of CO2 being removed. And then it is also goes through dehydrogenation, and hydrogen is removed, accepted by NAD, and we get reduced NADH being formed. Our 5-carbon compound is then going to be converted to a 4-carbon compound. When this happens, decarboxylation occurs again, so by the removal of CO2, dehydrogenation occurs, and NAD picks up the hydrogen and becomes reduced NADH. We also have substrate level phosphorylation occurring, and ADP and a inorganic phosphate gets donated from a donor molecule and produces 1 molecule of ATP. Dehydrogenation occurs again, and a molecule of FAD accepts that hydrogen and becomes reduced FAD. And dehydrogenation occurs again, where NAD picks up that hydrogen and becomes reduced NADH. We eventually get that 4-carbon compound with it, which is oxaloacetate, and then the cycle can continue again. So let's see what happens to all those products of Krebs cycle. So products of the Krebs cycle. We've got one molecule of coenzyme A, and that is now going to be used. It's going to be recycled back and used in the next link reaction. It's recycled back and used on the next link reaction. We have got our 4-carbon compound, oxaloacetate, oxaloacetate, and that is going to be used in the next Krebs cycle. We have got 2 molecules of carbon dioxide, and they are waste. We have got 1 molecule of ATP, and obviously that's going to be used for energy. At this point, our net gain, and this is of 1 Krebs cycle, can I just say? This is the product of 1 Krebs. So obviously, per glucose molecule, everything would be duplicated. So I'll put our numbers here if we're doing it per glucose molecule. It's per glucose molecule, it'd be 2 lots of 2 lots of that, 4 lots of that, 2 lots of that. So currently at this point, our net gain of ATP is 4. ATP, we've got 3 molecules of NADH, reduced NAD, and that is going to be used in oxidative phosphorylation, oxidative phosphorylation. And we've got, so obviously in per glucose molecule, that's 6 of those we've got. And then finally, the last thing we've got is 1 molecule of reduced FAD, which per glucose molecule, that'd be 2. And that is going to be used also in oxidative phosphorylation, which is our final stage of aerobic respiration. So, oxidative phosphorylation. This is possibly, let's just go back here. So, Krebs cycle, done. Last bit, yeah, oxidative phosphorylation. It's a messy one to try and draw, so just bear with me because I'm going to try and draw it out. Yeah, it's the final stage, and yeah, we'll go through it now. So oxidative phosphorylation is basically the part where we're going to make most of the energy, and it's where the energy carried by electrons from our reduced coenzymes, so reduced NAD and reduced FAD, is used to make ATP. So the whole point of everything, actually, these previous stages is to make reduced FAD, reduced NAD. Oxidative phosphorylation involves the electron transport chain and chemiosmosis. So it's why some people call it electron transport chain, but it's not. So I'm going to draw this out for you now. This here is our outer mitochondrial membrane. And I'm just going to draw electron carriers straight away. So what I'm doing is I'm just going to get the basics of this set up so then I can talk it through. Okay, nearly there. Just need to put in this and the other membrane, and then we're done. Oops. So we're still inside the mitochondria at this point. So that is our outer mitochondrial membrane. This area here is our intermembrane space. So we're between the membranes, intermembrane space. This is our inner mitochondrial membrane. And then this at the bottom is our matrix of the mitochondria. Okay, so what happens here is oxidative phosphorylation. So I'm going to go through how it works. What happens is hydrogen atoms are going to be released from that reduced NAD and reduced FAD that we've collected from all those previous stages. Okay, so what happens is we have NADH, and now I'm just showing you with one molecule of NADH, but obviously you know there'll be lots of molecules of NADH that we've got from before, and they're oxidized. So this is going to be stage one. Reduced NADH is going to be oxidized just to NAD, and exactly the same thing is going to happen to our reduced FAD. That's going to be oxidized just to FAD. Now, when these are oxidized, hydrogen atoms are released. Okay, so I'm going to use NAD as the example here. So 2 hydrogen atoms are released here. And what happens is the hydrogen atom is split. So I'm going to show it here for a bit. It's split into electrons and then hydrogen and protons. So split into electrons, and our electrons go up here. And it's also split into protons. So we're going to do them as hydrogen ions, and they're going to go here. Okay, so the hydrogen atom splits into protons and electrons. Happens for reduced NAD, reduced FAD, and this is going to happen lots for every single molecule, for every single coenzyme that we've got of NAD and FAD. But we're just going to talk sort of about it once. So basically, hydrogen atoms are released from all the reduced NAD and all the reduced FAD as they're oxidized to NAD and FAD. That hydrogen atom, as we've shown here, splits, and it splits into protons and electrons. Now, what happens is those electrons move down the electron transport chain. Now, these little weird blue circles that are drawn here, this is carrier 1, carrier 2, and carrier 3. These are the electron transport chains. And then these are electron carriers. And what happens is the electrons move down the electron transport chain, losing energy at each carrier. So what will happen is the electron will be taken up by this carrier. It moves across to the next one, moves across to the next one. And as the electrons are moving across each electron carrier, they lose energy each time. Now, this energy is really important because that energy that is being lost while those electrons are going through the electron carriers is going to be used to pump protons from the mitochondrial matrix into the intermembrane space. So we should have protons down here. Proton here, proton here, proton here. Okay, so as these electrons are going around the electron carrier. Let's pop it here. Electrons going around the electron carrier. Number 2, they lose energy. That energy is going to be used to pump these protons into the intermembrane space. So they are pumped from the matrix into the intermembrane space, and they're able to do this because the energy that's lost during the electron carriers allows them to pump those protons from the mitochondrial matrix into the intermembrane space. And remember that intermembrane space is space between the inner and outer membrane. So we did have lots of hydrogen ions down here, but we've now got a low hydrogen concentration now. Now H. So that's stage 4. Because they've been pumped from the matrix into the intermembrane space, we've now got a low hydrogen concentration down here, and we're going to have a high hydrogen concentration in the intermembrane space. So what we've formed is an electrochemical gradient. Just stick a few more hydrogen. So the concentration of protons is now higher in the intermembrane space. So I'm going to pop high H concentration up there. So it's now higher up there. So what happens is this forms an electrochemical gradient. So it's higher at the top than at the bottom. So because of this, our protons are going to want to move down that electrochemical gradient. Very standard. So are we going to do this by going back across into mitochondrial membrane and into the mitochondrial matrix? And it's going to do this by something called ATP synthase. They're going to come down here. This is going to be stage 5. We're going to go down this electrochemical gradient via ATP synthase. I'm just going to colour this in so you can see this is something different. This is ATP synthase. ATP synthase. Okay, so the protons, just said, got electrochemical gradient. We're going to move back to the across the inner mitochondrial membrane into the matrix, and it's done this via ATP synthase. And this ATP synthase is embedded in that inner mitochondrial membrane. And what this movement does is it drives the synthesis of ATP from ADP and the inorganic phosphate. So what's basically happening here is we've got ADP plus our inorganic phosphate, and it's going to make ATP. So basically, every time hydrogen passes through, ATP is made. So this process of ATP production is driven by the movement of the hydrogen ions across a membrane. And this is called chemiosmosis. So this whole section here is chemiosmosis. Chemiosmosis. Okay. And then in the mitochondrial matrix, we should now have a hydrogen ion. Let's pop it in. At the end of the transport chain, the protons, electrons, and oxygen from the blood combine to form water. So what then happens is the electrons, oops, let's keep the same colours going. The electrons are going to join all of this together. So we're going to have our half water and our hydrogen ions. Now this hydrogen ion has come from here, okay? Now I've made that really messy, so I'm just going to do that so you can see that's the same one going there. The electrons are going to join it together and we're going to make water. Okay. So oxygen is said to be the final electron acceptor in this process. So the final little bits about this. 32 ATP molecules can be made from one glucose molecule. So let's actually have a look at this and look at where each ATP is being made. I'm going to come back to this image in a second. So quick overview of this stage, which obviously is oxidative phosphorylation. Oxidative phosphorylation. NADH, FADH2 are oxidized. They release hydrogen atoms. The hydrogen atoms are split into protons and electrons. The electrons are taken up by the electron carriers. As the electrons go across each electron carrier, they lose energy. This loss of energy allows the hydrogens, the protons, the hydrogen ions, to be pumped across the intermitochondrial membrane into the intermembrane space. By doing this, we then get a low concentration of hydrogen ions in the matrix and a high concentration of hydrogen ions in the intermembrane space. This sets up an electrochemical gradient. This electrochemical gradient causes the hydrogen ions to then be pumped back into the inner mitochondrial membrane via ATP synthase. This is called chemiosmosis. Now, as the hydrogen ions are moving into the matrix of mitochondria again via ATP synthase, ADP plus the inorganic phosphates are forming ATP each time a hydrogen ion passes through. Finally, the oxygen from blood, hydrogen ions that have come from here, join together with the electrons that have come out of electron transport chain, electron carriers, and form water. We call water the final, oxygen is said to be the final electron acceptor. So the oxygen accepts the electrons, joins with the hydrogen and makes water. So let's look at those final ATP then. So let's look at what each stage makes and what happens. So oxidative phosphorylation, you know, makes ATP and it uses energy from the reduced coenzymes. What happens is 2.5, because we can't have simple numbers, 2.5 ATP are made from each reduced NAD. And 1.5 ATP is made for each reduced FAD. So I'm going to show you how much ATP a cell can make from 1 molecule of glucose, okay? And we need to remember 1 molecule of glucose produces 2 pyruvates, so the link reaction and Krebs cycle happens twice. So in glycolysis,
Rebecca, just
hiya.
sorry for interrupting. Yeah, hi. He's going a little bit uneasy because it has exceeded 15 minutes.
Oh, okay. Yeah, no, sorry, didn't realise.
So,